right. Hey everybody. We are back with another guest speaker session. Today we have Wisdom. Wisdom is a PhD candidate at University of Washington, supervised by Professor Ranjay, Krishna, and Linda Shapiro. His primarily project focuses on like large-scale multimodal curation and vision language specifically for medical AI. With that, he has like top publication at conferences like NeurIPS, CVPR, ICCV. He has a couple of awards as well. For example, NeurIPS Oral Award, UW Population Grants, and he has been part of the Microsoft Accelerating Foundation Model Research Award as well. Today, he's going to talk about distilling multimodals, pre-training evaluation from unstructured medical teaching videos. That's one of the papers that he has recently worked on. And over to you, Wisdom. Thank you so much for joining us today.
Yeah, thanks for inviting me. I'm gonna share my screen. Let me know, can you see it?
Yeah, we can see your screen.
Okay, perfect. Well, thanks everyone for coming in today. Thanks for the warm welcome and us. I'm gonna be talking about distilling multimodal training data and evaluation benchmarks from unstructured medical teaching videos. This is mostly with the end goal of developing spatially grounded vision language models for medical imaging. To start, I'm going to essentially discuss what the typical trends has been in the domain. So typically with recent breakthroughs in multimodal modeling, has come scale in both the data and the models. This is a challenge in the medical domain, as I think most of the audience for this particular talk would know. And we need to address 4 critical bottlenecks across the domain. First, we need scale in paired image text modalities. Second, we need diversity of connected modalities. So this can include stuff like varying views of the same anatomical structure or the same disease pathology, or varying modalities on the same patient. And this helps the models understand how different perspectives relate to the same clinical case. I think spatial localization of features is also very important, as models need to ground their understanding to specific regions in the medical image and not just recognize global patterns, as a lot of times the local structures and morphology and the relationships between them actually hierarchically tell a better semantic story in the high level towards like a diagnosis. Finally, we need annotations that connect low-level descriptions of visual features to high-level clinical reasoning and diagnosis. This hierarchical setup enables models to learn similar to how clinicians do. This kind of data set essentially would support a suite of downstream tasks, including classification, retrieval, medical VQA, localization tasks, and even generative tasks as well, right? Because you essentially have the suite of annotations that support a bunch of these kind of models, especially medical VQA. Now, while all of this is possible, the difficulty specifically in the medical domain it comes from the data privacy. There are strong HIPAA protections and institutional review requirements that make hospitals reluctant to openly share medical data sets. Also, the cost of annotation when injected into clinical workflows is enormous, actually. I believe it is more costly for doctors to additionally annotate more, give more annotations than is required for a report. And there's actually been studies that track and quantify that cost in terms of mental cost, in terms of the actual cost of time back to the patients, and it's a lot. And this cost also goes into the manual lab operations as well in collecting that patient imaging, right? So where can we get this data? Well, this is where YouTube becomes valuable. It serves as a unique resource for with 4 key advantages. First, it contains large sets of videos with frames and synchronized audio from various medical domains across various medical experts. It has many channels that review patients holistically across multiple relevant modalities towards the diagnosis. And this mimics the typical clinical workflow. Third, it has a platform that essentially forces narrators to point at features they're describing using their cursor due to the non-physical nature, as they are essentially on this platform where they can't physically, instruct or teach. And so they have to use their cursor as a source of localization. Fourth is the educational nature incentivizes presenters to provide low to high level descriptions naturally. And so essentially we've discovered a data source. But before I go into medical narratives specifically, I'd like to discuss a bit of prior work that informs medical narratives. So a few years ago, we started working on Quote 1M, which explored how to collect image text data specifically within one restricted domain. At the time, this was histopathology. I'll go into more detail soon on the different sections and how we curate the data, but quickly and more high level, we start by searching YouTube for relevant medical content. We extract histopathology images and remove duplicates and blurry images. We then obtain text with ASR, that's automatic speech recognition. We parse that text and correct it for medical misspellings or inaccuracies. And then we extract the medically relevant text. And finally, we pair everything together. This gives us images, medical text, and extra metadata like UMLS entities. And with specialized classifiers that we train for this particular work, we can also extract the magnification of the different patches, right? Because histopathology is a gigapixel scale domain. And so the patches that we see, we'd also want to know at what scale do they come from. Here we try to characterize the collected data. On top here, we see distribution across, or the distribution across subpathologies. We see that dermatopathology comprises a large chunk of our data set. I can say definitely that this cut came from 3 channels who had a very long history of teaching dermatopathology on YouTube. This distribution is you can see it further in the number of image text pairs that we collect. And you can see that other types of histopathology subdomains like cytopathology or hepatopathology, which are very similar and have very low counts within the data. And now, if you look at the UMLS entities that we curate from the text, we see that a lot of our text contain findings and neoplastic processes, ideas or concepts. They discuss cell or tissue or diseases and stuff like that. And finally, we display the most frequent terms in our corpus. Here I'm just showing a qualitative example of a rich expert level descriptions that we capture. You should notice how we go from the noisy text data to clean data, like for example, the cyst here is producing keratin, indicating keratinous cyst. We also capture like I said in my prior slide, we also capture the slide magnification, where the larger microscope icon here essentially means more zoomed in. So how useful is this data? Well, we fine-tune a modified clip on the data, We leverage the base clip VIT as the VIT base as the image star. We also leverage the base clip text star, but we also have experiments with an adapted BERT text encoder that was pre-trained on medical PubMed data. This particular text encoder has a much larger context length, which is better suited for medical text. Our model outperforms all baselines in aggregate with strong performance in both zero-shot retrieval, where our modified text outperforms on our held-out set, and in our linear probing test, this is more supervised where we train like a linear head on top of the model. We see high, improved performance over baselines on our patch level test sets, which demonstrates a transfer of our length representations, right? To finally visualize the attention map, we see that QuiltNet, which is the name we gave to our model, aligns medical text well, in this case, for histopathology to specific image regions better than CLIP model. A great example is the fatty tissue here should be describing this white out region. And in CLIP, it's all over the place, but for our model, it's well localized, right? So far, we've established a new source of data. We've shown it's useful for fine-tuning foundational models, but we're still restricted to one medical domain, histopathology, and we still don't collect pairings between various medical imaging domains. We don't curate low to high level textual descriptions. We also don't collect any spatial annotations that ground the text to localized image regions, right? This brings us to medical narratives, which addresses some of these limitations. Essentially, we expand to 12 imaging modalities, including X-ray, CT, MRI, ultrasound, ophthalmology, and more. We collect the same rich expert narration, but this time adding in spatial traces. Some examples you can see here, we'll see more down the line, to the text to localize the image regions. In parallel with our work, actually, PMC and MedTrinity 25 million both curate large training datasets. However, PMC lacks spatial annotations and medical videos, and MedTrinity lacks novel images and video. They also lack human annotations, and as they caption, they mostly caption the images from pre-existing data. So they lack both the novel images, and they also lack human annotation, right? In contrast, medical narratives is unique with 4.7 million samples from both videos and articles across 12 domains. We introduce novel images, videos, expert level text, spatial traces, and also have multilingual support. So I'm going to go now into the specifics of how we curate the data. I'll start with the video pipeline, which has 5 stages. First, we search and filter for narrative style videos using a suite of domain classifiers. So for search, what we did was we built a database of medical keywords spanning all 12 domains. We built this using different medical ontologies, and then we use that to search YouTube. This helps us to identify channels and videos with relevant content, and then we can sweep through every video with our trained classifiers for each modality to then narrow down or filter out videos that we really care about. We essentially narrowed the videos that we care about. Again, these are what we call narrative style videos, by leveraging chunks of videos that have stable backgrounds. We identify those stable backgrounds using SSIM. Obviously, those stable backgrounds are on medical images rather than on, say, people's faces or some other content. And then we classify if a video is narrative, depending on how much they dwell on the medical image content across the entire length of the video. These shrinks are number of videos curated greatly, from 748,000 to about 74,000 high quality narrative videos. So to essentially collect the frames and the videos that we do, we identify two types of regions in a video. Specifically, we identify extended stable regions that also provide long stable backgrounds for cursor trace extraction. And then we also identify short stable frames. So this would be an example of a long stable frame, and this would be an example of short stable frames, where we perform the duplication and then we remove redundant content while preserving the visual diversity within the medical images that we do have. For text, as you can imagine, the text is very noisy. As humans aren't robots, and so even in the educational nature that we do find in most of our videos, you do have a mix of both medically relevant to the images and non-medically relevant to anything at all in the audio transcriptions. But first, one of the main issues that we had was actually having a medically fine-tuned ASR model, which we couldn't find or which couldn't train, because that would require a different type of data that was even harder to curate. So we essentially used an off-the-shelf ASR model, and we followed that by correcting the transcriptions and then extracting the medically relevant text. Specifically, we built a 4-step text denoising pipeline to clean the noisy ASR transcript. First, we essentially use Rake for our error extractor here. We use Rake to pull out key medical phrases, dropping out stop words, and then we validate those phrases using UMLS, which is a database of medical text, medical entities, essentially, using entity linking. And if there's something that doesn't match, we treat it as likely misspelled. So, for example, with this system, we are able to extract words like spirochetes and submucosa. Third, we prompt an LLM with in-context examples and also this sort of errors that we've been able to find to both correct those errors and catch any other errors that we must have or may have missed or didn't flag, right? And then fourth, we only apply the corrections that parse that particular verifier again. This is to avoid any sort of hallucinated fixes, right? So at the time, LLMs weren't as good as they are now, and so they also would make certain mistakes, and we don't want to hallucinate those mistakes back into the data. The outcome of this is that we essentially ignore filler words, and we automatically focus the LLM on medically relevant terms. And then from this clean transcript, we're able to use the LLM again to separate out two things. First is the general medically relevant text, and then second is ROI text, where this is just text that we curate from the subregions of the ASR, where the main narrator is essentially pointing out specific parts of the image using words like look here, look there, stuff like that. Next, we tackle the traces, which we didn't in quote 1M. Spatial traces here are extracted from long stable chunks within video. For each chunk, we compute.
Is it okay with you if I ask a quick question?
Of course.
How do you know what they are saying? So if you have the medical terms, but you still need to know, maybe they are saying it is not this. Maybe they are comparing two different things, and the image that they are looking at is not the same as the text that they are pointing at. So you have to have an understanding of the text as well, correct?
Yes, and so that's why we leverage the language model rather than building any sort of like heuristic method. And so we prompt the language model to specifically extract the medically relevant text, right? And so there's also the medically irrelevant text, like you said, where, for example, they might be discussing a particular case, but also sort of recalling a different case, but we still have the current medical image in front of us. And so we instruct the model to essentially leave out anything that's not medically relevant to the current image. So this LLM essentially has a summary of the history and also can see the raw text, right? Yeah. So that's how we.
Thank you very much.
Yeah, please stop me anytime for additional questions. Happy to take them. Yeah, so for the medical traces, we essentially extract them by looking at the stable regions. And what we do is we compute the median frame within a stable region in the pixel domain, and we subtract it from every other frame. This essentially helps us to pinpoint where the cursors are, as we essentially hypothesize that the only things changing in those stable regions are the cursor movements, right? Because again, it's a pedagogical sort of environment where one narrator is essentially trying to teach others. We then apply thresholding to reduce the noise, because you can still have several noise because of the video domain and maybe codec encoding and stuff like that. And then we take the maximum value to capture the mouse cursor points. These points or traces are then encoded as normalized XY coordinates relative to the image size. And for alignment, we use those identified stable regions to determine the refined text to assign to the frames. We typically pad the region's timing to avoid missing any details. And then we use keyword matching to combine nearby chunks, ensuring that we have sort of temporal coherence between visual content and narration. For articles, because we also do capture data from articles, we leverage open PubMed set, similar to prior work, actually, but crucially, we filter much better. To start, we searched over 5 million PubMed articles. Within those, there were about, I think, 23 million figures, and we filtered out about 1 million medical figures from over 200K articles. We essentially use the same set of classifiers that we leverage for the video pipeline here. Next, we fine-tuned an object detection model specifically for detecting subfigures in compound medical figures. We leverage LLMs to then split the captions. This avoids, again, very similar to the last question, any sort of heuristic approach that might fail when the captions aren't like enumerated in the same way that whatever heuristic you can sort of come up with might be able to fail at. Finally, we use the pre-trained OCR model to extract the subfigure IDs and pair that to the captions. For cases where the IDs are more abstract, so for example, you can have cases where an ID for a particular subfigure says right or left in the captions. We use the bounding box positions of the subfigures to essentially match with subcaptions based on the spatial relationships that we can extract from the bounding boxes between, like, for example, between two bounding boxes, we can tell which one is on the right, which one is on the left, which one is top left, which one is bottom left, stuff like that. Our final data set contains over 700,000 unique images from 74,000 videos. We total about 4,000 hours, and we're able to extract about 1.6 million image text pairs and also 1 million image text pairs with spatial traces. We also curated over 4 million UMLS medical entities and also over 100,000 video clips. From articles, we extracted 1 million figures with 2.7 million subfigures across 12 imaging modalities. And the radar chart here essentially shows our coverage across different submodalities. So here's an example of a CT image, right? The transcript here describes the frames over time, localized to specific regions in the image. And the extracted medical text essentially captures the key anatomical descriptions within the particular video. Again, here's another example that captures paired views within the MRI domain with synchronized narration as well, similar to the prior example. Finally, we also have this example for X-ray that demonstrates that our trace extraction and cursor points at this sort of like demonstrates that our trace extraction is very accurate or matches the text accurately. As you can see, the cursor points at specific points match the text descriptions. This actually was something we found very interesting, as it means you can essentially do a lot more. This can essentially enable a lot more sort of downstream work. For example, tracking text to point location, modeling, stuff like that. Similar to Quilt, we train a foundational clip model called GenMedCLIP. GenMedCLIP, not particularly very novel name, but on zero-shot classification across 11 medical modalities, our model achieves the best average performance across various benchmarks within each medical domain. So within each medical domain, for example, X-ray is a bunch of different benchmarks. And on average, we perform well on CT, dermatology, histopathology, mammography, and ultrasound. On retrieval tasks, GenMedCLIP outperforms all baseline, and specifically it outperforms BioMedCLIP by 26%. We also perform ablations to discern what data source, since we have a mixture of data sources, contributes the most to performance gains. And we see that training on video data gives an average of 66%, while articles gives an average of 12.7%. This demonstrates the complementary value of both data sources. But this also quenches any sort of questions that may arise from the noisiness of the images from the video site. For linear probing, on average, GenMedCLIP outperforms all other models, including BioMedCLIP and PubMedCLIP, with over 2.8% and 1.9% respectively. And finally, we demonstrate the segmentation utility of the collected traces using Scribble prompt. I believe you also had the Scribble prompt earlier in this series. Here we use the minimal amount of traces from our collected traces, and we achieve high quality segmentations of anatomical structures like brain tumors or knee joints. This shows that the spatial grounding from our trace annotations transfers effectively to denser segmentations using iterative segmentation models like Scribble prompt. So far we've discussed, or we've shown that we can collect a diverse set of modalities. We can ground our textual annotation to the images. However, we've yet to collect hierarchical textual features or textual annotation towards like medical reasoning, or leverage the spatial traces to guide any sort of vision language understanding. This prompts the next work I'm going to talk about. And so just a backstory to essentially ground sort of the timing of the projects. While developing medical narratives, we prioritized the histopathology domain, as we already had stable chunks from the first work, Quilt 1M. And so we leveraged the narratives to work on Quilt Lava and released it even before medical narrative. So Quilt Lava was released CVPR 2024. Medical Narratives was released NeurIPS 2025. So essentially, Quilt Lava leverages low to high level textual data all localized to regions of the images to tune a vision language model, enabling spatially grounded medical text generation towards image captioning and diagnosis. So to start, how do we essentially leverage the localized narratives, right? We do this by injecting the localized narratives into the text modality. To add in the spatial annotation to our text for training, we group spatial traces into meaningful regions through spatial temporal clustering. Specifically, we apply exponential decay to the spatial coordinates. This is to ensure points that are closer in time are essentially preferentially clustered together. We then select the number of clusters that we wanted for each stable region based on the number of words in that stable segment. Then we assign the words to the cluster based on the distance to the temporal midpoint of the stable region. Finally, each cluster is represented essentially as a bounding box. So while this system isn't perfect, what we did to validate this was to do an internal sort of like clustering, human clustering, and then we essentially use a different set of methods to essentially perform this clustering, and this performed the best on that. Next, we collect hierarchical low to high level textual annotations for instruction tuning. Specifically, we collected or curated several types of instruction tuning sets. We curated conversational QA pairs. This is for basic image understanding. We also curated detailed descriptions for comprehensive analysis. Then we created complex medical reasoning for diagnostic thinking, and finally, iterative abductive reasoning QA pairs for multi-step clinical problem solving. Essentially, this captures the full spectrum of simple perception to high-level reasoning. To curate both the complex medical reasoning and iterative abductive reasoning, we essentially leverage the extracted diagnosis and supporting facts. And so to do that, we essentially focus on videos. For these two specifically, we essentially focus on only videos where you had solely one whole slide image being discussed that belonged to one specific patient. And to curate the iterative abductive reasoning traces, we essentially pitted two models together against each other. One model, which you can call the student model, or LLM in this case, essentially had access to the images, descriptions, and was tasked with reaching a diagnosis and collecting supporting facts from the images it had access to. And the teacher or the guide LLM was essentially tasked with guiding that student towards the diagnosis without essentially giving out much more than hints, which we essentially collect from the supporting facts for that particular case. So this system essentially simulates like an educational system and the ability to reason progressively towards a high-level diagnosis. In all, we extracted over 160,000 image caption pairs, and we filtered this down to over 114,000 based on the word count, because we wanted higher word count for our instruction tuning sets. And for reasoning-based instruction sets, we manually reviewed over 4,000 videos and selected over 2,000 videos. Finally, our Quilt Instruct comprises of about 107,000 question-answer pairs across all four instruction tuning sort of like subdomains. So interestingly, due to the educational nature of the data source, we're also able to extract out the benchmark. So we developed Quilt VQA with over 1,000 expert quality image QA pairs. And unlike synthetically derived question-answer pairs, educational videos essentially provide additional information and we're able to identify questions using the question marks in the actual narration. And after quality checks, this particular benchmark avoids any sort of issues from prior benchmarks like wrong answers or inconsistent pairs and so on and so forth. We contribute a better evaluation benchmark, especially for histopathology. We also consider localization of features via visual prompting using red circles. This is very similar to clinical practice where radiologists and histopathologists or pathologists, as we call them, would essentially use black markers or stuff to circle out regions of interest. This is to test models whether they can attend to visually highlighted regions when answering questions. Our benchmark includes, on a high level, both image-dependent and general knowledge questions. The image-dependent questions require visual analysis, and the general knowledge questions test medical understanding. We train a lava architecture. So essentially, we use that data to train a lava architecture. In stage 1, we use quilt data. In stage 2, we use our instruction tuning sets. And at the end of the day, we end up with a model that can perform visual question answering well. So for evaluation, we evaluated quilt lava in 2 ways. First, with GPT as a judge, because we wanted to see how well the model followed instructions, how much helpfulness it provided versus other models, and the accuracy of the answers. We also evaluated the model on histopathology-specific VQA benchmarks. On the GPT-based alignment evaluation across 14 subpathologies, quilt lava variants beat the baseline. Our lightweight recipe here, which we only trained on 40K samples to start, improves over lava and quilt lava, and medical lava med by 16 and 7% respectively. We then scaled instruction tuning data and we trained for even more iterations, and we perform even better over time against lava med. On the VQA benchmarks, we see a consistent sort of improvement over prior baselines. For open-ended questions, we outperform by 4% on average, and for closer questions, we outperform by 9%. Qualitatively, when asked questions like what's the connective tissue in the visual image, quilt lava correctly identifies the pericardium, while other methods misidentify the tissue completely. And so one other emergent, well, maybe not emergent because we sort of trained for it, is that it can also localize in words where that particular feature is. For example, in this case, it says it's the upper left quadrant, which is accurate. We also see improvements in multi-turn conversations. It provides comprehensive description here when tasked with describing the image. It provides really good descriptions of the lymphocytes and eosinophils in the tissue. And when asked for a diagnosis, given the supporting facts that it has extracted, it's able to suggest inflammatory response, but correctly also notes that a definitive diagnosis requires additional context. This demonstrates appropriate medical reasoning, right? So in summary, we've presented 3 major contributions: Quilt-1M, a large-scale histopathology dataset from YouTube. Second, we've discussed medical narratives expanding to 12 medical modalities with over 4.7 million samples, with diverse parent spatial traces, which are grounded to the text. And finally, we discussed quilt lava, showing how localized narratives essentially enable strong visual question answering, performance, and appropriate medical reasoning. So thank you. By the way, code, data, everything is all open source. Thank you so much.
Thank you so much, Wisdom. Really nice talk. The floor is open for any questions anybody has. Feel free to ask any questions. Yeah. All right, maybe I can start. I have a question. So like you're using the LLM for the ASR, right? So like, how do you make sure like how to tackle like in particular the hallucination problem in that? Do we have like a super factual checker on top of that or something like that to make sure the hallucination is not that much? And what particular LLM are you using specifically for that?
Here, yeah. For the first workload one, we used the best model at the time, which was, I believe, GPT 3.5. And then for medical narratives, I believe we used GPT 4.1, which was the best earlier in the year and I think late last year. And specifically, the way we avoid any sort of hallucination of words is that we do a double check, right? So we essentially task the LLM here with words we know for sure are not in any medical sort of like database of terms, but somehow appear here. And the LLM corrects those, right? But we also task the LLM with flagging down other errors. So for example, in this case, it's also able to flag down Sehosa and turn this into Serosa, right?
Yeah.
Now the problem is that Serosa is not a correct word either, right? So we then need this verifier to then check for the correct ones, right? So that's how we avoid this sort of hallucination, because we have this sort of double check. If we had only one check, then yes, hallucinations can just fall straight into the text. But yeah, because of that, we're able to avoid that. I hope that answers the question.
Yeah, thank you. Thank you, Mr. Yeah, any other questions from our
I have a question. So you mentioned in the GenMedCLIP, right? So the experts basically use segmental approach to avoid errors. So I wanted to know if the clip itself inherently learns the systematic way of reading the image, or is it primarily focused on identifying the most salient clinical features described by the instructors?
Sorry, could you repeat the question?
Okay.
Yeah.
So yeah, I was just saying that for the GenMedCLIP,
Mhm.
basically you highlighted that the experts use segmental approach to avoid errors, right? So I wanted to know if the model itself learns a systematic way to read the image, or is it primarily focused on identifying the most salient clinical features described by the instructors?
I see. I think maybe there's a miss. So GenMedCLIP is simply a clip model, right? And in training GenMedCLIP, we don't use the grounding, the spatial grounding from the traces in any way. So that's why I'm still misinterpreting the question somehow. So I'm wondering what you mean specifically by the attribution back to the text. I'm not sure.
Yeah, so the thing is, I was a little confused with I thought there was some kind of ground truth that is being given, and I wanted to know if the model inherently learns that way of reading the image so that it aligns with the ground truth. Or maybe I misinterpreted my understanding, I guess. So yeah, that's fine.
Yeah, I really wish I could give you a better answer, but just to speak more broadly, maybe I hit the maybe I somehow hit an answer to your question. GenMedCLIP is trained specifically on just image and text. We showed earlier that training on this on just this fine-tuning on just this data, you do have better performance against prior benchmark that even trained on larger sets, right? Like BioMedCLIP. with QUILT-1M. We also showed that when you visualize sort of the attention heat maps between the two modalities, you have better sort of localization of feature importance to the actual regions in the text, right, against CLIP. And then we realized that we've done all this work to curate, medical narratives and QUILT-1M, but we haven't shown that it's usable for any sort of segmentation task. And so that's when, a bunch of iterative segmentation models started coming out, including Scribble Prompt or UniverseEgg or MultiverseEgg. You can name a couple, NN iterative. And so we showed that you can essentially take the traces as the positive points, and then you can randomly sample negative points outside of those positive points, and you can essentially curate much denser segmentations. And then we realized, okay, that's well and good, but these segmentations are also very noisy, right? So how do we use the actual traces that we have in the best way towards understanding? And that's where we started working on QUILT-Lava, where we clustered those points into B boxes and tied those B boxes to several text regions or text annotations. And then we have a data that then looks like this, right? Now from this, we're able to curate a bunch of instruction tuning sets. And then those instruction tuning sets are then used to train QUILT-Lava. And then QUILT-Lava has this sort of emergent ability to localize its text into like regions, as we've shown in this particular example. So I hope that was helpful.
Yeah, that's fine. Yeah, thank you.
Thanks.
I also had another question about the cursor. So I think I saw in the paper that the paper notes that identifying cursor traces is very difficult due to varying speeds and styles. So how did the algorithm handle instances where the instructor might draw a circle like a region versus simply pointing or hovering over? So did one style provide a better training signal over the other?
Yeah, so the way we sort of handled varying sort of narration speeds and styles is to essentially adapt to number of words instead, right? Because given a particular, say, 5-second, 10-second window, you can have, varying speeds. And so we use the number of words rather than just taking, just like sampling maybe a number of clusters based on the points. We use the number of words to figure out what the clustering numbers should be. And specifically, the way we did that was we did sort of like an internal test where we took a bunch of videos and we essentially created clusters around the points, and then we assigned those clusters to like a brute force like middle point of the text, right? So we say, hey, this first part is this particular cluster of points. Draw a B box around it. Hey, this is another cluster of points, and then we assign text to it as well. And then we started developing several heuristics by which we can make the same sort of alignment to human sort of like alignment as well. And so what we realized is if you're going to create those clusters, the most important thing is to align the temporal points that are close together, because you can have pauses in when the cursor moves, right? And those pauses over time, they might not be visible spatially, right? Because you just see all the points at once. But if you just look at the timing, you can cluster those points as one and then cluster the next set of points as one. So that that exponential decay in the timing of each cluster point essentially helps also to inform how many clusters there are, right? So yeah.
Thank you so much.
You're welcome. Thanks for the question.
All right. Anybody has any other questions? Feel free to ask. All right. If nobody has any questions, let's end the session here. Thank you so much, Wisdom, for taking out time and presenting today. This was
Thank
a
you.
very helpful session. And for people who want to reach out to Wisdom, Wisdom, can you share your email or anything? How can people reach out to you?
Oh yeah. Yeah. Can you still see my screen?
Yeah, we can still see.
Oh yeah. So email is right on this website. Now I don't specifically. It's just wisdomik at uw.edu or at cs.washington.edu. Yeah, please feel free to reach out for any questions, collaboration, or whatever. Thank you so much.
All right. Thank you so much. Thank you, everybody.